This is big picture anatomy with a view of the eye in sagittal section. And what we're looking at is the eyeball as if we've cut it right across, right down the middle. And then over here a close-up of what's happening with the cells in the retina. If we take a slightly closer look at the eyeball, what we see is that we've got the outer sclera, or white part of the eye, that actually forms the eyeball itself, made out of tough collagenous connective tissue. That sclera, in fact, what it is really an outpocketing of the dura mater of the brain, which extrudes out into the orbit through the optic canal where the optic nerve leaves, and it extrudes out and forms the sclera around the outside of the eyeball. Just internal to the sclera is the very dark choroid layer that has lots and lots of melanocytes, giving it that hard, dark black and also highly vascularized look when looking into the eye itself. Now at the very front of the eye, what you're going to see is that the sclera becomes the cornea and is this very crystal clear glass-like part of the eye that you're like looking right into, and that light initially enters into. Just behind the cornea is going to be the pupil opening, which is surrounded by the colored part of the eye, or what's called the iris, with the pupil, which is just an opening right in the middle. It appears dark black because you're actually looking in on that dark choroid layer at the back of the eye. And then behind that is the lens, the football-shaped lens that's held in place and is given its shape by the ciliary muscles or bodies that surround the lens. You have to think of this as like a donut in 3 dimensions, going out of the screen and into the screen, entirely surrounding the lens. The same would be true for the iris, of course, around the pupil opening. So the lens divides the eye into an anterior segment between the lens and the cornea that is filled with a very liquid material called aqueous humor, a very crystal clear fluid, and then the posterior segment, which is filled with a more jelly-like, also crystal clear material called the vitreous humor. So let's take a look at what happens when light actually comes into the eye and how we form a visual image. So here's our light. It passes through the cornea, which rounds out the light so that when it falls on the back at the retina, it will be rounded out in that same shape. The iris or pupil opening actually regulates how much light is going to enter into the eye. The pupil itself is formed by the iris, which is made out of smooth muscle, and it's colored by melanin. The thickness of the layers of melanin determines the various levels of eye color that we get. So the light passes through the pupil opening. Depending how much light there is, it might be very tiny if there's a lot of light, or wide open if there's not too much light. The lens then fine focuses the light, and then it passes through the posterior segment, the vitreous humor, to land on the back of the retina. Now, the retina itself is a series of photoreceptor cells and neurons that are laying on the choroid layer, and that translate those photons or light rays into a neuronal signal that is then sent to the brain through the optic nerve. All of the axons of the optic nerve converge at this point here, which is actually that blind spot in your visual field. If you trace it right out from there, there's actually no visual receptors there, and so we have the blind spot where that your brain regularly fills in with whatever it expects to be there. If we take a closer look at what's happening in the retina, that's what we've got over here, what we'll see is that as the light passes onto the retina, it passes through these neurons and to the photoreceptors, where what are what's typically called the rod and cone cells. And what we see is the photoreceptors have this internal cell membrane filled that's got a protein in it called rhodopsin, and that protein is responsive to light. It changes shape when the photons of light hit it. That causes the cell to trigger a neuronal response. It synapses with what's called a bipolar neuron, and those bipolar neurons do some processing already of the various photons that are falling back here, and then sends that neuronal signal as an action potential through what's called a ganglion cell, through the axons of those ganglion cells to the optic nerve and back to the brain. You've got rod cells that are sensitive to any color light and would give more or less a black and white image, and then we've got cone cells that are tuned to particular colors of light. There are blue cone cells, red cone cells, and green cone cells. All right, then, just to summarize, what we've had here is a sagittal section of the eye, and we can see how light would come through the cornea, through the anterior segment filled with aqueous humor, passing through the pupil opening formed by the iris, regulating how much light comes into the eye, and through the lens, which fine focuses the image, regulated by the ciliary bodies that stretch out or allow the lens to ball up due to elastic fibers in the lens. The visual image then lands on the back of the eye, where it's interpreted by the photoreceptor cells that send a action potential or neuronal stimulus via the bipolar cells to the cone cells that go back to the brain via the optic nerve, which leaves the eyeball, creating a blind spot where there are no photoreceptor cells. That then is a view of the sagittal section of the eye from big picture anatomy.